And thank you all. We're going to have the first panel discussion from this morning's session, and I think you can all agree it was a fascinating morning. We've learned that asking and solving questions can lead to important results, such as what you've just heard here now. And we'll have the session with this morning's speakers, Dr. Tim Hunt, Professors Langer and Emmanuelle Charpentier. And please, when you ask a question, remember again to identify yourself and your affiliation. So, we are ready for questions. We have a couple here that have come in through the live stream. So I'll start with Bob Langer. What advice would you give to young scientists who are just starting out?
Yeah, well, no, really, I mean, actually, my advice is kind of simple, I suppose, to follow your passion. You know, I think you certainly heard that yesterday and today. I mean, people, you know, really talked about that. I mean, I think it's just important to do things that you love. I mean, you know, it's not about, and I hope also doing something you love that does some good for the world, but just things that make you happy and things, I mean, I guess.
But don't you think it's important to also to find a place where you feel comfortable? I mean, to work with people who you would like. And
absolutely.
I mean, that's super important, I think.
I completely agree. I mean, that I probably should have added that. I mean,
or maybe you thought that was obvious.
No, I don't. Actually, you know, it's funny when students come to me and ask about, you know, jobs and what they're going to do, I often say, like, the most important thing to think about is who they're, you know, who are they working with.
Who you're going to be working with.
And who's your immediate boss, even, to really even understand that.
Yeah.
Because a lot of times, you know, people go and you do interviews and you don't know that. And so I think the immediate boss of where somebody works is probably, but it's also hard to know that, you know.
Well, I found it much easier when I had students in the lab. I mean, if I was, you know, in Cambridge, they were super bright students, but many of them I didn't want to work with because I just didn't like them. I mean, you know. Well, I don't mean I didn't like them. I thought some of them were much cleverer than I was, and it was better they go and work somewhere else. You know, that kind of stuff. But it was important. I mean, you know, occasionally, I once remember we took on 3 graduates, and for some reason, I never really figured it out. They just didn't get along with each other. Well, it was poison in the lab. It was terrible.
Well, that's a whole other issue, you know, and I'm sure everybody here, you know, that's run a lab, I mean, that's the thing I hate the most, you know, when you have conflicts between people and how you deal with that. That's
very bad.
Awful. Well, I mean, nobody's...
Let's hear from...
and if I may, this is what we understood yesterday. Chemistry is everything. And actually, for me, this has always been about chemistry among people.
Yeah.
And I guess you feel it even when you look for labs. You know, you have to listen to your guts, and you understand whether there is a good chemistry or not. I mean, everyone you know went through it. Sometimes even you meet people, and you're like, oh no. In life, it's the same. And I think the chemistry is very important among the people.
Thank you. I have some other questions from the live stream, but I think since we have such a long line and an ambitious program, let's go ahead.
Hi, I'm Werner, a student from Katedralskolan, and I'm wondering if you think that the biological development is a new age for humanity. An example is that we had the Industrial Revolution. Now all of the products we have are from industries, and also we had the technological development, and now we are surrounded with technology. So will it happen the same with biological mechanisms and organs, that we are surrounded with biological mechanisms?
Who wants to start?
You start.
Yeah, I think it goes very fast. I mean, as you know, when I was starting as a student at the Pasteur Institute, this was in the beginning of the 90s. So the World Wide Web was not existing. So we had to go to the library to, you know, to enter into a kind of horrible computer, some the search for papers, and then we had. I mean, I was lucky. I was at the Pasteur Institute. There was a huge library, but you needed to go to the books and take the big books and photocopy everything. So, and very fast the PDFs came. But the idea at the time that that the world will be as it is today. That I could call from a watch. I mean, I even don't think that we were thinking about it. We were thinking that it was belonging to James Bond movies or Star Trek. But so I think there is a tremendous development, and it's only 20, 30 years. So I believe indeed that it's going to go exponential, and that and it's going to go faster and faster. That's why we really need people, young people like you, to really follow the path.
You had a slightly negative part of it, that the development might not be to our what we really want, and sometimes you could encounter situations where you need to take your fate in your own hands, and for instance to start a company. I think we all agree that the present grant system is not always the best one.
All right, I think we're going to move on to the next question since we have quite a few to answer.
Yes, all right. Hi, my name is Tage Rosenqvist. I'm a PhD student here at the university in applied microbiology. And I was just questioning on a bit more personal note. So a lot of the speakers at this symposium have had some significant public opposition from various groups, creationists, anti-vaccine activists, GMO skeptical movements, and other skeptics in general. So I was just wondering, how do you all, on a personal level, deal with that sort of attention? And do you find it easier or harder to deal with that sort of criticism as compared to criticism from your fellow scientists? Maybe you're...
Yeah. It's... I mean, I think they're both. I don't like either one, but how do you deal with it? I mean, I think in science, of course, you can hopefully write articles, that show that, you're right. I think when when you deal with some of these activists and stuff like that, it's a whole different thing. I mean, and of course, it's not a community that you live in the same way you live in the academic world, but that's certainly out there and has, certainly can have an effect in all kinds of ways. I mean, and some of it leads to things that aren't pleasant. I mean, where people threaten you and stuff like that. Mm-hmm.
Yeah. I think the truth, though, does tend to come out in the end. Mm-hmm. And, I mean, there was another nice quote from the great Schrödinger. He says, "Science is a game, but a game with the reality." And at the bottom, there is that reality which we all deal with and which tends to keep scientists honest and true. And heaven help you if you fake things, because you'll always be found out.
Okay, thank you. Could we move on to the next?
Hello, my name is Mohamed. I come from Procivitas Gymnasium in Malmö. First of all, I want to thank all the speakers today for the amazing talks. And also in the public, there's a lot of students, high school students, with teachers. And some of us are interested about the question. That is, how can we change school curriculum to fit science better?
Yes. Yes.
Have more fun, less exams.
Yeah.
Standing ovation.
But on the other hand, I actually had a funny experience of that. I was a postdoc at the Albert Einstein College of Medicine, and the powers that be decided that they would do away with exams for the medical examination, for the medical training. It was a disaster because none of the very bright students in those years passed their board exams. They couldn't become doctors. So, they quickly went back to having it. So I'm afraid exams are things we have to live with. But teaching to the exam is awful. Oh, dear, oh, dear.
Any other comments?
Well, I'll give a little bit. You know, I was thinking, you know, when I got moved into that chemical engineering department, they wanted me to teach thermodynamics. And boy, that was hard because, you know, a lot of what the curriculum was like 18th century stuff. And students, you know, they kept saying, well, why do I need to learn this? So I always sort of thought two things were important. One is actually what you said, to make it fun, you know, somehow, which isn't easy, but
no, especially in thermodynamics.
Exactly. but the other thing was to try to make it relevant, you know, and I think that if you come up with ways. So, for example, I didn't get into this in my talk, but there's different mechanisms, chemical mechanisms like osmosis, as an example. So people have designed osmotic pumps where you can get absolutely constant release, and so you can come up with examples like that. And I did some demonstrations, so I think when you make things relevant, that also helps.
Well, I think that's what he's asking is how do we make it relevant
with real examples, like just like what if there's a way to take the kinds of things you heard this morning and yesterday, you know, from and put them into like questions or things. I mean, I think that does make it relevant, but it depends on the course. I picked thermodynamics deliberately because that was hard for me, you know, and I struggled with that.
But I think, I mean, I was always better at practical things.
Sure.
You know?
But that's kind of what
showing people actually. They were the great things.
Well, but that's why I meant with an osmotic pump. You can
see it, and then you have to figure out what's going on.
Yeah, but you can put a dye in, and you can put dye.
See it, watch it in action. Yeah.
Yes, thank you.
Thank you. Next, please.
Hello, my name is Omar Hassan, and I am from Gothenburg Kunskapsgymnasiet. And my question is directed to Emmanuelle Charpentier, professor. Your achievements have been a great inspiration to me and many others. Given the remarkable progress in CRISPR-Cas technology, what do you see as the most promising applications in the life science and medicine? And what advice do you have for us as a young students to help advance and learn from this technology?
Yes, so to reply to the first question, actually my answer will be like it was when the technology started to be applied, actually. You know, we already see that the technology can be used to treat genetic disorders, but de facto it's only on a very limited number of diseases.
But people can cure thalassemia, is that right? People can cure thalassemia, for example.
Yeah, so beta thalassemia and sickle
I mean, that's
cell
pretty
disease.
impressive. No?
Yeah, it's very
Yeah,
impressive.
very.
It's very impressive, indeed. Specifically because those diseases are extremely painful, and that the expectancy of life is also limited. And so it's here where, you know, when you have a genetic basis of the diseases, you can always for sure apply a genetic technology, but it's not as, let's say the blood disorders were also chosen as a first target because it's easier to target
blood cells are very easy to get.
Because you know the idea is to take them from the patient and transfuse them back. So this was also the idea to use those diseases as a first proof of concept. But you have multiple genetic disorders, even more and more, that are discovered year after year. So I think really the biggest development of the technology to be foreseen in the future is just more application of CRISPR-Cas to treat more diseases. Because the genetic background is not always the same, so you cannot always apply the same trick, to cure one disease versus another one. So this is really what I expect to see clearly, whether directly as a direct medicine or indirectly. And for sure, was a challenge that is really to develop the technology for in vivo applications, where here, everyone focused a lot on CRISPR and the precision of CRISPR, the safety of the technology. But I think very early on, at least me and others have understood that the challenge is more delivery technologies that really make the difference for the technology like CRISPR. Then advices, I mean, I guess I gave some advice already this morning. And I think I will say exactly what was said. We are scientists, so we try to encourage you to do science, but you don't have to be scientists. I think the most important is that you carry on with what you would like to do. You have actually also, science is also now very broad. I mean, a lot of people call themselves scientists, which I will not specifically think they are scientists, but maybe they are. I think the encouragement is more with regard to the fact that it's a unique opportunity to fulfill your curiosity, and if you're creative, to also fulfill your creativity. This is at least the reason why I'm a scientist, and I think it's all about people as well. I feel, at least my experience has been this. It's really about people, and even founding a company. It's having interacted with people in the past and knowing the people with whom I will co-found the company, etc. So it's all about people, and you have a chance to enrich yourself so much by having the chance to meet scientists from all over the world. And I think I wish that the students also will understand very early on, the high school students, that being a scientist is not being behind your bench and pipetting all your life. It's more complex than this. At the end of the day, we're a little bit, when we start a lab, it's like we start our own startup. We can do what we want as long as we can gather the money to do what we want. It's a unique opportunity to be really independent. It takes a while and it can be scary, and for me, I thought it was impossible. And my dream was maybe to open a field. I was always fascinated by my mentors who had opened their own, find their own niche and open. I thought it was impossible, and I figured out that it was going step by step and through failures. But I think it's so important that high school students understand that scientists are also communicators, or managers, you can fulfill a lot of what is in you which you don't know exists, and you will understand bit by bit that it exists, and it allows to reveal so many facets of your personality. That is actually what I found the most interesting, and you learn so much from the people, and also from the young people, which challenge you constantly. So I think it's great.
It's a very social activity, actually, much more so than the popular sort of...
Yeah.
Okay. Thank you very much.
Thank you. Go ahead.
Hey, my name is Arian, and I represent the Swedish National Science Team. And I have a question for Professor Langer. So I've read one of your papers on these sort of star-shaped oral capsules for the delivery of anti-malaria drugs, especially in developing countries.
Right.
So I had a question: whether you think that oral drug delivery could ever replace more intrusive conventional methods, especially for vaccine administration in developing countries?
Yeah, well, I hope so. I mean, so just a couple of things. Actually, I have some I didn't put that in. I actually have some demonstrations probably in one of my bags. But so the oral one, the whole idea of what he's asking about is, and there's two different things I'll mention quickly. One is we came up with some materials, and you can sort of compress them into into something small enough as a capsule, but it's got what's called shape memory. So when the capsule dissolves in the stomach, out this thing pops. But we designed it in such a shape that it's so big that it can't pass through the pylorus, at least for a period of time. But it's so open that food can pass. And then we made a part of it that will ultimately dissolve with a degradable system, so you can make it last 1 week, 1 month, and so forth. So that's actually now in phase 3 clinical trials for schizophrenia. That's another company we started called Lindra, and it's in phase 1 trials for the malaria thing. The other thing, though, that we did is that we also, and this has been published both in Science and Nature journals, came up with a pill you can swallow that's got a little needle in it, except this time the needle's made of the drug. So we've actually delivered, and so the point is you swallow it, the needle sort of pops out, but it's doesn't hurt you. I mean, it's actually been tested in 65 patients now. And so it pops out, and, we've used it in animals to deliver, insulin, to deliver messenger RNA and so forth. I mean, obviously, neither of those are going to be out right away, but I think someday they will be, or something like them.
Thank you.
Wow.
Okay, next, please.
Yeah, hi. So my name is Elin, and I am a master's student here at Lund University. And it was what Timon said about how sometimes you just have to do the experiment. That really resonated with me because we can discuss results until we're blue in the face, but at the end of the day, we have to do the experiment. But I would love to hear more about your thoughts about this balancing act between reading literature, reading articles, analyzing data, planning experiments, and all these other things that aren't the actual lab work, and how to kind of, find that balance between what to focus on and when to do the experiment.
Well, I think chatting to people is really important, actually. It's you just never know when you're going to get a good idea which is really going to open the door. Sometimes it's reading something. Sometimes it's talking to people. Sometimes it's hearing a talk.
Conferences like this.
Conferences like this. I've every which way. You just never know where the next. Because very often, I mean, in my experience, you spend an awful lot of time being stuck, and you're sort of stuck in a conceptual rut. You're not. I mean, I realize in retrospect, many times you think, gosh, how could we be so stupid for so long, right? And so you need something, go somewhere, talk to somebody, do something different. It was funny, I mean, when I used to go and spend my summers in Woods Hole, which was great because it's a sort of summer camp for biologists. It's a very wonderful, wonderful place to be in, whereas I always found American scientists, rather competitive on the whole. They somehow lowered their guard a little bit in Woods Hole, and that was
great,
nice.
Yeah.
But, there were a lot of people, advisors who said, oh no, you can't possibly take 2 months off to go on a course. This will be a disaster for your PhD. But every time I spent a student there, it was amazing the energizing effect it had on them because they realized that they were the equal of their peers. They also realized that in a week you could learn some new technique and be, not the master of it, but good enough to get on with your work and to open new horizons. So I think sort of shaking yourself out of convention is terrifically important. It's really easy to, when things aren't going very well, you drag your feet into the lab and you drink lots of cups of coffee instead of doing the experiments or reading or talking to anyone. You get very depressed, and then it's a sort of death spiral from which you have to escape.
Emmanuelle?
What Tim said is so true, but I guess for you the challenge is that you have so many publications in nowadays.
Yeah, how do you make sense of that plethora?
And you have to, to figure out what is crap and which is 90% and what is not crap.
Well, it's not all crap.
It's not all crap. For CRISPR, for example, it was easy. I knew all the papers that were published. I knew them by heart. I mean, it was maybe 30, 35 papers, and I knew them to know what is known and what could be, found, new. So it's always finding the black box, the question mark or the question that people maybe did not answer. And I think it's important to know because you need to know the field and what you can bring, that is interesting to the field. But
knowing the people is also
challenging.
very helpful, actually. I mean, did you know all the people in?
No, but at the point I didn't know any of them.
You didn't know any of them, so it was just reading.
And yes, and so I could think freely and my own way. So sometimes it helps to not know the people.
Because in my case, I mean, when I started in the cell cycle...
And sometimes it helps to know people, surely.
Yeah, when I started out in the cell cycle, I mean, I was completely ignorant. I was completely aware of my ignorance, so talking to people. But then I quickly became aware that actually the number of people who knew anything worth knowing was very, very small. I mean, almost fingers of one hand, people like Mark Kirshner and Paul Nurse and co.
But me, it was different because it was really an emerging field. So these were a bunch of scientists coming from structural biology, enzymology, phages, who had, come together to CRISPR just 10 years before, and just a couple. So it was a very, very small community.
Yeah, well, it was the same in the cell cycle. I mean, there was almost, I mean, can you believe how almost nobody worked on the control of the cell cycle in the early 1980s because nobody could see how to do it, right? And so the number of people who did, the number of sane people who you could talk to, were very, very smart and very, very few. And we all worked on different things, which was helpful. You know, some worked on flies, some worked on frogs, some worked on yeast. And so there was really no competition. It was all, you know, clue. You never knew where the next clue was going to come from. It was really fun, really wonderful, actually. And then the medics joined in, and it was hell.
I want to add one thing. You study a lot, and you come up with questions, heaps of questions. Then you begin to think. You come up with a hypothesis of your own. So the next step is, how do I test this hypothesis, verify or falsify it? So that's the next step. And then you are into the system.
Okay, thank you all. Thank you. Our next question, please.
Hi, my name is Ville. I'm from Nösnäs gymnasiet. I go in year 2. I have a question for all of the scientists. I want to ask, how did you choose your area of science? Could you, for example, have ended up in physics, maths, or maybe, I don't know, literature or something? Why are you specifically in your area?
Well, I'll start because I have a terrible answer. But you know, so in high school, I was good in math and science, and I was not very good in anything else like English or French. So my dad and my guidance counselor said, well, you should be an engineer. I mean, I really didn't know what an engineer was. I thought they ran railroad cars. But at any rate, you know, I didn't know any better, so that's why I did that. So I majored in engineering. And then when I went as an undergraduate, my first year, I thought the courses were really hard. I didn't like any of them other than chemistry. So I picked chemical engineering. I mean, which is, like I say, not the. I always hope people will do much better than me. But that's how I got started.
What about you?
With me, actually, my mother will. When I chose my lab where I will do my master's, this was at the Pasteur Institute. I was working in different labs before, but she told me, it's funny, when you were 12 years old, you came back one day saying that, I will work at the Pasteur Institute. So I thanked her to not have told me.
That's great.
I thought that she should have told me before, but having said this, you know, in France there was a pressure that, you know, the good students are the ones going to mathematics, etc., at high school and science. So since I was a good student and I wanted to remain good, I thought that this was what I had to do. But I was interested in other types of sciences, also human sciences. Then after, I thought that actually human sciences, I kind of understood that it was a little bit fuzzy as a career. And then maybe science, it was, you know, there were more options actually with regard to, you know, being independent and doing something also meaningful. After I chose microbiology, I think it was again the gut feeling, you know, something that I became interested in. So, but it was really step by step. When I was 15 years old, I wanted to be at some point a detective and work in a monastery. I was very focused on this, and then after I understood that actually it's like being a scientist. You're in a monastery, and you're a detective. You just, you know. You're just trying to solve the enigma of life. So it was maybe not completely far-fetched.
But there's a big stochastic factor. You don't really know. It could be this or that. And
yeah.
Sure.
Yeah.
So before we go on, I'm just going to say that we'll end the questions with the line that we have now because we have a tight schedule today, but there'll be chances to ask questions during the breaks. Okay, so after you, that'll be it. But Tim, do you want to say?
Well, I just did very well in a biology exam when I was about 12 years old, and I realized that I was, it just sort of, that way of thinking came naturally to me, and then after that I never worried about a career. I mean, it's just a question of, did you pass the exams? And it was easy. I was very lucky, I think. I knew people who were much cleverer than I, and they never knew what they wanted to do, and they were very unhappy.
Okay, thank you. If we could have the next question, please.
Hello, my name is Emily and I'm a third-year student at Hvitfeldtska gymnasiet, and I have a question for the Nobel laureates, and that is, what do you think is the greatest benefit that your discovery has brought to mankind?
You start.
Yes, so first
Yeah,
of
I think
all,
she has actually benefited mankind.
yeah, we are Nobel laureates, but you know, they have to choose 3 scientists. In my case, they chose 2, Jennifer Doudna and myself. But you always feel that, you know, who should have, maybe, I mean, you have always other colleagues who have also contributed, you know, prior to your work. Yeah, actually, as I said, CRISPR-Cas is really making the difference, opening new avenues in medicine and also in agriculture, for example, the possibility to engineer plants in a precise fashion, differently than the techniques that were primarily used to modify through GMO technologies and also using pesticides, etc. So it makes a difference. And in medicine also I did not talk about, but you have also transplantation, you have other types of technologies that benefit a lot from the CRISPR-Cas9 technology. So it's really large applications, and I believe that when it comes to, let's say, allowing the people to have a perspective when it's about, you know, eating food for those who do not have access.
It's obvious.
And
But what about you, Tim?
It's obvious.
I don't think I've benefited mankind at all. Actually, I mean, well, I don't know. I mean, it's better to know than not to know, surely. That's the main thing.
Okay.
And cell division is kind of important.
Well, there was quite a body of people who did think he benefited mankind, so...
He's modest.
We'll take the next question, but...
Thank you.
Thank you.
Yeah. Hello, my name is Clara and I'm also from Hvitfeldtska gymnasiet. I've heard a lot of the speakers during these last 2 days talk about diversity among scientists. And I would love to know your thoughts regarding diversity and also the actual measures that can be taken to promote diversity in the science community.
Who wants to start?
I mean, maybe I can say there is a bit of a focus on the word diversity nowadays, where actually for me it has always been diverse, and the diversity came extremely naturally. I think us as scientists, we never cared so much where the people were coming from and you know, their sexual orientation, etc. It has never been any of the criteria. And now it's a little bit, I feel that this diversity that may be a problem in other fields, you know, has now come to the field of science, and we feel that we need to then show that we are diverse and reply to the statistics of also the number of women in science, etc. Well, it does not work like this. It works because, you know, we have scientists coming to us. We cannot, you know, we cannot influence the type of scientists who will come to us, whether they are more female or male, or whether they come from that country or that country. But I guess we are very open. We always took into consideration the diversity. And I think we can be proud of it and continue to do it and stand for, I think, a field that really did a lot, continues to do a lot, and take really diversity completely as a natural criteria. And in a way, yeah, this is my opinion.
Okay. Well put.
Yep.
And a lot of efforts have been done as well to facilitate, you know, this I have to say because you're young people, and what I appreciate a lot in the young generation is that also the men take, you know, their family responsibilities a little bit differently. So every aspect that was difficult for a female scientist because of, you know, not having a proper, you know, childcare, etc., applies now to male scientists who also claim, you know, we have a problem, we want also to pick up our kids, and we have the same issues. And so I think that it's what I like. It's all those issues that were maybe more female and less male now are becoming the problem of everyone. And I think it allows also to less, how do you say, stigmatize on, one category. And I think the scientists are doing really a lot of efforts to promote diversity in that case.
Okay, thank you.
Well
Thank you
said.
for your question. If we could have the next.
Hello, thank you very much for all your presentations. It was very interesting to hear. My name is Eric and I'm from Lund University, the Department of Biochemistry. And I'm currently in 2 groups, and my question was originally towards Professor Langer, but I think all of you will have some great input on it. I've been doing a lot of lab work in diverse fields, and 1 year ago I developed some kind of a new protocol method, and right now I'm in the patent mud and trying to develop this into a company. And my question is, for a person in academia, what's your biggest tip for a person to try to develop a company out of an idea you have?
Yeah, well, I think it's hard to say. One thing, I think if you're doing things in the medical area, generally it is good to have a patent in it, to file for a patent. But I also think it's good to get advice from people who have done it before. Maybe you find mentors at the university or in the local community who, especially if they have some experience in that area, that can give you advice. And I mean, myself, like I'm a scientist, but a business or an engineer, but the business world is quite different. So to me, creating a company, if that's the goal, it's really a team effort. So you want to get good business people as well as good scientists. I realize I did more than one tip, but and there are probably better tips than what I just said. But those are 3 things that I think about.
Thank you. I think we'll move on to the next question.
Hi, so my question is for Professor Langer. So your group recently published a paper on a new type of nanoparticle, a metal organic framework called ZIF-8. It was able to encapsulate and deliver part of a SARS coronavirus to spike protein, while also acting as an adjuvant.
Right.
So what are your thoughts on ZIF-8 as an alternative vaccine carrier to like conventional liposome nanoparticles?
Yeah, well, first, I'm impressed that you're up to date. That came out pretty recently. So, I think, of course, all these things are early. But, and there's a couple of things, and this was kind of mentioned yesterday. You want to make sure that the things work well, but you also want to make sure they're safe in humans. So so things like that, and I hope it does lead to something someday, it's going to be a lot of testing to get it to the point where it could be used. But absolutely, someday there'll be better nanoparticles, there'll be better adjuvants. I mean, I'm sure with all the things that people are doing, I'm sure there'll be very significant improvements, different routes of delivery and so forth.
Thanks.
I mean, immunology is a bit of a black box still, right? That's the problem.
Yeah, that
very
is
complicated.
Extremely.
And not perfectly understood by any manner of means. I mean, all this adjuvant business, I mean, it's very mysterious.
Well, you're right, and I think that's a whole exciting area to understand a lot
so
better.
there's still, don't worry, there's still lots to be found out. There's still tons of stuff we don't understand.
Exactly.
Even in sort of quite well-worked areas, you don't. The trouble is, books, you read these textbooks and it makes everything sound very pat and wonderful and now, ta-da, this is what we know. And they don't like to dwell on what we don't know. So they tend to sort of brush that under the carpet.
That's right.
All right, thank you. So our last question for this session.
Yes, hello, my name is Masoud and I study at Ystad Gymnasium. And I noticed that I noticed that you mentioned that finding funding for a startup was kind of a big part of getting things going. And so I was wondering if any of you have implemented the idea of using AI for the purpose of cutting expenses. And if so, how has that worked out in your research?
Boy, oh boy. Well, I'm not a great believer in AI myself.
Well, you could get your grant application.
I suppose you could write a grant application, but it would look awfully plain vanilla, wouldn't it? I mean, You know, the trouble is that it's all based on the past, as far as if I understand it right. You know, it can't, and AI can't do experiments either.
No.
Can it, or can it?
Well, actually, one of the questions for you was they were asking if AI will do the work for us in the future, and can it?
I'm unconvinced. I mean, when my wife and I spent 6 years in Okinawa, and when we came back, we found that the internet wasn't working at home. So we had to call the engineer to sort of wire us up. And, you know, I talked to all these chatbots. Twice I sat at home all day and nobody came, you know. And again I did it. And it wasn't until I found a human being to talk to that the problem was solved in an instant. You know, it's bizarre. So I mean, but everyone says, oh, well, it's only in early days. But the early days to me are not terribly impressive.
Okay, go ahead.
But then the question was AI and expenses.
If it was going to help
or
be able to monitor and make more efficient expenses and budgets.
That seems very unlikely.
You mean the expenses associated to the grant funding?
Associated with the experiments, is that true, or grants?
To have your to have AI do parts of your research or job. So that you can cut expenses, I guess.
Oh, to save that way, I guess.
I guess, I mean, at least when we do science, we spend money. We have to ask for money. We spend money. We have an administration with bookkeeping, accounting that became more and more digitalized. I mean, we think more and more that bookkeepers, who are now human beings, and accountants will be less and less human beings and more AI behind. So in a way, I will foresee, yes, indeed, that at least you will have more digitalization and AI to at least, if you have a grant application, I guess you have to write it, the scientist who has to write it, but how do you say that will help you to anticipate the budget you need, based on the type of experiments you want, and as well will, be able to put together all your expenses and put a schema all well done with regard to the human resources fees, etc. That will be at least helpful, and that will come from digitalization.
Any type of efficiencies will already start saving you money.
Exactly.
Thank you.
All right. Well, thank you all. Thanks to the panel. And now we have a little bit different format. We have a special guest during the lunch session. So for those of you who are extremely hungry, I ask you to leave now. And otherwise, we have a speaker who will speak to you, and you can ask some questions, and then you'll have time still for a quick lunch. So if you'd like, you can take your seats, and I'll introduce our next speaker. Thank you.